study Koch's postulates. This was given by Robert Koch, right? Named, obviously, as usual, the scientific, the person of science. So this is a great importance in medical microbiology and advancements in understanding the etiology, right? So you'll understand what etiology is, because this is basically what it is. The postulates, there are actually 4 postulates described below here. These give the criteria to establish the causal relationship between a microbe and a disease, right? So how do we know that this microbe is responsible for this disease, right? And how to experimentally prove it. It's not a hypothesis, it's not a general observation. And these postulates are basically criteria that you have to justify in order to establish this relationship. And this reaffirms the germ theory of disease. Okay, prior to that, in earlier times, think about, you have no knowledge that even microbes exist, right? You might notice that people with poor hygiene, maybe in the more slum areas, you see more disease. People are generally suffering from diseases, and you see a lot of abundance of that, right? So it might be fair to assume that something is coming from the rotting matter, right? And that was the miasma theory. So miasma theory of disease believed, or it was actually now an obsolete hypothesis, that foul smelling vapors or foul air, smelling air from rotting organic matter causes diseases, right? And there were many other misconceptions, and we didn't really properly understand disease. So what the germ theory says is that a specific microbe causes a specific disease, and that's what the postulates go on to affirm. Okay, so the first here is, first of all, you would see a manifestation of this disease in a certain host, a susceptible host. Okay, what you're going to do is, the first postulate is that this disease should appear in all, this microbe should appear in all cases of this disease. Okay, in a particular disease, a particular microbe is appearing in all cases. Basically, this emphasizes the fact that a pathogen is consistently associating with hosts of a particular disease, right? And this goes on to imply that this will be absent from non-diseased individuals. Secondly, you're going to isolate and propagate a pure culture, right? If you know how you create a pure culture, take your sample, you dilute it, you streak plate it, there are many methods, right? And then you'll see development of individual colonies. You pick it up, and now in this new petri plate, you'll just grow that one colony. It will increase in number, and this will be your pure culture. So why this step is important is for the next postulate. The next thing you do is you introduce this isolated microbe into a healthy susceptible individual, right? And this is basically the experimental infection step. And what should happen, what should be the result, is that this should reproduce the disease. Okay, so you took your from your deceased person, you take that microbe, you grow it in a pure culture. This would ensure that disease is caused just by that single microbe and not by a mixed population of contaminants, right? So upon introducing it into the susceptible host, you should see a reproduction of that disease, that particular disease. Now the fourth is kind of reaffirming it. You re-isolate it from this new experimentally infected host, and you would see the identical organism. You would have extracted the identical organism, right? So first step, you see that disease manifestation, you isolate, grow it, you will experimentally infect a healthy host, and you re-isolate and confirm the presence of that organism. This is affirming that the disease that manifested here was due to the presence of the organism that we inoculated. Okay, and what he did was he worked with mycobacterium tuberculosis. So there are TB lesions, right? These are called reservoirs. This is the place, this is the primary site where the pathogen is multiplying, right? So these appear in tuberculosis on the skin. So he extracted it from here, he isolated and grew a pure culture, then he introduced it into a susceptible host, and after that re-isolated. This confirmed that tuberculosis was caused by mycobacterium tuberculosis, right? Now the importance of this is that first of all, it confirmed the germ theory of disease, which has been revolutionary in medicine, and thereafter we've been able to determine the causative agents for several infectious diseases such as anthrax, cholera, and this has also helped us in disease diagnosis, development of vaccines. And if you look at it, this was the base for us being able to develop vaccines and antimicrobials. Once you're able to identify, only then you can characterize it and you can target it. And last point, it introduced an experimentally scientific approach in infectious disease research, right? Because when you perform these steps, or you work by these postulates, you're following a very reproducible protocol, and also this relies on lab evidence, not any, it's not general understanding or observation. You have lab evidence and causation analysis. And if you look more far into it, also the basis for epidemiology, understanding how infection spreads in a population. It has allowed us to prepare for such epidemics, right? Employ preventive measures, develop vaccines and such. And there's also some limitations, because we talk about growing it in a culture, but we know viruses need live cells in order to grow. There are also some species that don't grow that well in artificial lab cultures, artificial environments. There are also ethical limitations, because generally you're doing all these experiments in lab animals, right? But there are some diseases only manifest in humans, but you can't go and just infect a person with a potentially deadly disease, right? And an important point here is that the first postulate says that it should not appear in diseased people, but there are many diseases that have carriers, right? They're asymptomatic, they don't show any effects, or they don't show symptoms, but they are still carrying that disease. So that kind of disproves, doesn't fall into the first postulate. Also, there are many diseases that are caused by one causative agent, right? It's not always one agent causing one disease, right? A lot of effects can be induced. There is, for example, Streptococcus pyogenes, right? Causes skin diseases. It also causes rheumatic fever, and The other way around also, one disease also has many pathogens working together for the manifestation. And sixth is the opportunistic pathogens. It doesn't take into account that, because a lot of microbes are present and they're actually kind of in symbiosis, right? In the normal microflora, they don't cause a disease, they're not pathogenic. But immunocompromised, or if they are present in not their niche, not their normal habitat, then they can lead to diseases. And a popular example is Candida albicans, right? Okay. Moreover, so now in today's time, we understand a more accurate. Just to be more accurate, we go by the molecular Koch's postulates, where we're not talking about the entire organism. Instead, we just consider the virulence genes. Virulence is the how much damage is caused by a pathogen. Okay, so these are virulence factors. They can be toxins, primarily toxins. If it's because of the pathogen, then the toxins do the direct damage. So, for example, you can consider that. So we basically consider that presence of this gene leads to disease manifestation, and if you were to get rid of it, there would be a reduction in the virulence, right? Now, obviously one gene isn't responsible for virulence, that's why there's a reduction. To get rid of all of the virus genes, there will be no manifestation of disease likely. But again, if there are antigens present and all that, you'll still have certain kind of, you might be febrile, you might be in a fever state. It's basically all for Koch's postulates. If you want to go over this again, here's a quick summary, right? Koch's postulates describes the criteria, or is the criteria, to establish the causal relationship between a microorganism and a disease. This proves the germ theory of disease. And we keep saying infectious disease. So just to be clear, a lot of people believe infectious means it passes from one person to another. No, infection, like when you use a disinfectant, you're not making sure that it doesn't pass from you to another, right? It's basically infection of the body. Okay, infection is when a microorganism, or not just always a microenvironment, any pathogen, right? Because we also have protozoa and we also have helminths, right? You have tapeworms. Those are also pathogens, but they're not microorganisms. That's just to specify. So infection is when such a pathogen enters the body and leads to manifestations of disease, multiplies, it evades the immune system. Okay. The other term is contagious when we're talking about passing from one person to another. So infectious disease is basically diseases that they're not like genetic basis. There are other ways you can get disease, right? Lifestyle, might be some imbalance in something, okay, damage. But when it's induced because a pathogen has entered your body, and it's due to an external pathogen, then we call it an infectious disease. So the four postulates being should be present in all cases of the disease. It should be isolated and grown in a pure culture, reintroduced into a susceptible host, and then re-isolated and identified. It should be the identical organism.